Imagine you're underwater, somewhere off the rocky California coast. It's quiet down here, except for the gentle pull of the current and the faint crackle of life in the reef. In the shadows, an octopus extends one elegant arm into a crevice. The suckers gently press against a cluster of eggs. She pauses. Then, without hesitation, she plucks one egg free and tosses it into the sand. Why? That's rude. How could she know, just by touch, which egg would never hatch? This isn't magic. This is neuroscience, and a little help from some very, very tiny friends called microbes. Welcome to Mapa Brain Lab. I'm Mayuri Patel, and today we're diving into a study that was just published this past summer from the lab of Nicholas Bellono at Harvard University. They've discovered how the octopus arms taste the world and how microbes secretly guide those arms to make life or death decisions, like how to find their next meal or toss out an egg. Humans have been fascinated with the octopus for so long, and so have I. Whether that's because of their remarkable intelligence, the way they interact with the environment, their shape-shifting camouflage abilities, or the almost alien way they move, octopuses constantly challenge our understanding of what animal minds can do. Their nervous system is radically different from ours. Most animals, including humans, have a centralized nervous system, a brain and spinal cord that coordinates everything. Octopuses, however, diverged from humans and other vertebrates over 500 million years ago. What that means is that we have a common ancestor, but we split and evolved differently over millions of years. But despite this, they've developed a nervous system that is highly intelligent. They evolved a unique system. While we have 86 billion neurons with most of them in our brain, an octopus has around 500 million neurons with 2/3 of their neurons in its arms, not in the central brain. Each arm can sense, process, and react independently, which means they can grasp, explore, and taste without waiting for instructions from the brain. Now, for years, scientists have known that an octopus's arm don't just feel, they taste. These arms are covered in hundreds of powerful suckers, up to around 2,000 in some species. It might sound strange, arms that can taste, but when you think about it, that's just another version of what all sensory systems do. Our senses are basically how we make sense of the world. They help us pick out the details that matter, from recognizing a friend's face in a crowd to tasting our favorite meal. And when we look at animals like the octopus, with their totally different sensory systems, we start to realize there are countless other ways to experience reality, ways that can teach us something new about how all brains, including ours, perceive the world. So yes, the octopus really does taste with its arms, but not the way that we do, where our tongues pick up molecules dissolved in saliva. An octopus's suckers detect chemicals that cling to surfaces, like the traces left behind by microbes, like bacteria or prey. You know those dining in the dark experiences where you eat blindfolded and rely on texture and smell to guess what's on your plate? Imagine taking that even further. Imagine tasting with your hands. For the octopus, that's everyday life. Every reach, every grab, every brush of a sucker is a sensory experiment, a way of feeling the chemistry of the world. Now, long before the study we'll discuss today, researchers had noticed special sensory cells packed inside the suckers that respond to chemical cues, not just touch. When an octopus explores its surroundings, it's essentially sampling the world one sucker at a time, detecting the difference between smooth rock, rough coral, or something that might be dinner. Earlier experiments hinted at this taste-by-touch superpower, but the exact receptors and molecules involved were still a mystery. Wait, what's a receptor? Receptors are special proteins found on the surface of cells that act like locks. They're designed to detect and respond to specific signals. When the right chemical fits into a receptor, like a key in a lock, it triggers a response inside the cell. And molecules are basically everything you can touch, taste, or smell, like sugars, amino acids, and fats. These are the kind of chemicals or molecules that receptors can recognize and react to. Scientists knew the arms could sense certain hydrophobic chemicals, ones that don't dissolve easily in water, but they didn't yet know how that worked. That part was still waiting to be discovered. So here's the big question. How do those arms actually know what they're touching? What's happening at the microscopic level that lets an octopus taste with its skin? That's where the new research comes in. Scientists have finally figured out which receptors are doing the work and what molecules they're responding to, and it turns out the process is even stranger and more sophisticated than anyone expected. Now, any good experiment starts with a strong hypothesis. A hypothesis is like a scientist's what if, a testable idea about how something works. It's not just a guess. It's a roadmap for finding answers that can be tested with experiments and data. It usually begins with an observation, and Bellono's lab observed two key things about the octopus. The first observation was that they often use their arms to find hidden food to eat. Imagine blindly digging through your pantry for a tasty treat and knowing exactly what every snack tastes like. For the octopus, this ability could be life or death, as many things could be decaying or toxic in the hidden crevices of the deep ocean. The second observation was when a female octopus is caring for her eggs. She stays with them, cleaning them with her arms, fanning them to give them oxygen, and guarding them from predators or harmful microbes. She doesn't leave to hunt or eat. Her whole focus is keeping those eggs alive and healthy until they hatch. And this behavior is called egg brooding. So let's recap. The two key natural behaviors of octopus arms that researchers found were foraging, or searching for potential food, and egg brooding, when the mother cares for her eggs. Now they could build a hypothesis. What if octopuses are using chemicals that are released from microbes like bacteria that are present in every surface they touch? Great. Hypothesis done. But how do you actually test this? Well, we know that the mothers are touching eggs, and one food that octopus love are crabs. So why not just look at what kind of microbial communities cover the eggs and crabs? Microbial communities are basically a mix of bacteria and other microscopic life that leave behind chemical fingerprints that the octopus can detect. Maybe what the octopus is sensing isn't the crab or egg itself, but the microbes living on it, each surface carrying its own chemical signature. So the lab went to work. First, they took live crabs and decaying crabs and checked out what's covering them with a scanning electron microscope, powerful enough to zoom way into these microbial communities. Surprisingly, the live crabs didn't have a ton of microbes, but the decaying ones were completely covered with different types of bacteria. That was a clue. Then they looked at octopus egg casings, ones that were mothered and others that were rejected. Under the microscope, the bacteria themselves looked different. The microbial coats on mothered eggs versus rejected ones had distinct shapes and textures. Additional experiments of these microbes showed that they were distinct from other objects in the octopus's environment, like the tank or their den, confirming this hypothesis that specific microbiomes on the surfaces of their food and eggs might guide octopus behaviors. But of course, they didn't stop there. We know that microbes can secrete or release molecules, and they asked whether there are specific molecules from the crab or egg surfaces that could be detected by the octopus chemotactile system. Remember I mentioned the suckers have receptors? They're actually chemotactile receptors. We'll call them CRs. They're tiny locks, and the molecules released from the microbial population could act as keys. When the right key touches a receptor, it sends a signal to the arm. This is edible, or this egg might not survive. So we know they are there, but can the octopus actually detect them? So they isolated the microbes, separating them from the mix, and then cultivated them, meaning they grew each bacteria in the lab to see what it was and how it behaved. They took these bacteria from the tank, the den, the suckers, different crabs, and egg casings. The researchers collected the liquid the bacteria were growing in, basically the chemical soup the microbes released, and filtered it down to just the small molecules. Remember, it's the molecules that the bacteria release that the octopus arms might interact with. Now stay with me. To figure out if the CRs on the octopus suckers would respond to these, the researchers cloned them. And I'm sure you've heard of the word clone, but what does that actually mean? They copied the receptor's DNA and inserted it into lab-grown cells, kind of like installing a new sensor in a test system, so they could test which molecules they had just filtered from all the bacteria could activate the CRs. So there were 2 CRs they cloned, and one of them, CRT1, was activated by distinct molecules, but the other had almost no activity. Another clue, CRT1. In fact, different molecules triggered the CRT1 receptor in different ways. Some caused a strong response, while others barely activated it. Now, the paper also dives deep into other areas we won't touch on today, including the specifics on the CR structure and how they bind the molecules, and broader analyses of how microbial chemistry influences the binding. Those are fascinating, but for this episode, we're going to jump to my favorite experiment of the study. So far, the lab identified microbes all over decaying crabs and these rejected versus mothered egg casings. We also know that they release chemicals that CRT1 on the octopus sucker responds to. But all of these experiments were in a dish, outside of the living, behaving octopus. And one thing you'll learn about scientists is that they are bold, and they don't stop there. They want to see it in action to prove that the same sensory magic actually happens in the real octopus. Fueled by curiosity and creativity, the lab designed a pretty clever experiment to test their idea. Let's start with the crabs. There were certain chemicals released by the bacteria covering the decayed crabs that activated the CRT1. So why not cover crabs with this chemical and see how the octopus reacts? Bingo. They did exactly that. They ordered mimic crabs, or plastic toy crabs, straight from Amazon and soaked them with these microbial chemicals, basically mixing the mimic crabs with the chemicals for 25 minutes. They also made a control batch of these toy crabs, soaking them with a neutral lab chemical that the octopus suckers don't respond to. Then, they dropped each crab into the tank and watched closely for 60 minutes. Observation is a scientist's superpower. What they found was amazing. The octopus touched and explored each crab, but only the one covered in the neutral chemicals triggered engulfing behavior. That's when the octopus pulls the crab under its web like it's capturing real prey. Remember, both crabs were just plastic toys, but the octopus could still sense the difference in the chemicals and decide which one felt or tasted like food, rejecting the fake crab that was covered in the decaying crab chemicals. It was a striking demonstration of just how finely tuned their chemical senses really are. Driven by the same curiosity, the team came up with another experiment, this time focused on the eggs. They wanted to know if those bacterial chemicals also influence how mother octopuses decide which eggs to remove. Now, they didn't order fake eggs. This time, they made them in the lab using a soft, jelly-like material called agarose and mixed in either the microbial chemicals or a neutral control solution. Once they cooled and hardened into little spheres, the team dropped these false eggs into the dens of brooding mother octopuses and filmed what happened. The mothers inspected the eggs with their suckers just like they would with the real ones, and the scientists watched how long it took the octopus to reject the eggs. They found that the fake eggs coated with the chemicals from the rejected egg casings were rejected significantly faster than the neutral solution, showing that the octopus arm could sense these chemicals and guided its behavior. Both of these experiments are what scientists call sufficiency experiments. That means the researchers were testing whether the chemicals alone, without the real crab or the real egg, were enough to trigger the octopus's natural behavior. In other words, could these microbial molecules by themselves make the octopus act as if it were sensing real prey or real eggs? If the answer is yes, that's strong evidence that these chemicals are sufficient to cause the behavior on their own. And the answer was a resounding yes. The arms weren't guessing. They were reading microbial chemical cues to make critical survival decisions. What does this mean? Every organism perceives the world through its sensory filters. For the octopus, microbes shape that perception. Chemical signals aren't just background noise. They are information the octopus uses to act. Senses are integrated with microbes, not hardwired alone. For humans, this resonates with ideas like gut-brain interactions and the ways our environment and microbes can shape behavior. It suggests there may be unseen microbial influences on our own sensory perception that we've only begun to uncover. Findings like these challenge how we think about intelligence, decision-making, and the very biology of sensation. Now that we've discussed what this study discovered, let's pull back the curtain and take a quick peek at the experimental details that made all of this possible. To help you follow along, I've put together a glossary of key terms so you don't get lost in the scientific language as we dive a little deeper. So the initial discovery of which microbes are responsible for chemical cues coating the crabs and eggs were done by profiling the microbial communities using 16S rRNA sequencing, which identifies conserved ribosomal RNA genes. This revealed distinct microbial signatures between the mothered and rejected eggs, as well as between fresh and decaying crab tissue. To test if these microbial metabolites could act as specific chemosensory signals, the team isolated dominant bacterial strains from each condition and cultured them separately. They then collected the cell-free supernatants and fractionated them by molecular weight and polarity, helping narrow down which classes of compounds, small polar molecules or larger hydrophobic ones, were responsible for activating the receptors. These extracts became the input for the electrophysiological assays of the cloned chemotactile receptors. This systematic workflow allowed the researchers to directly connect microbial identity with sensory receptor activation. Now, once the team identified candidate microbial strains and their secreted metabolites, the next step was to understand how these molecules physically interact with the octopus CRs. CRT1 was inserted into a DNA vector and introduced into HEK293 cells, or HEK293 cells, using a method called the Sleeping Beauty transposase system, allowing the team to create a stable cell line that expressed the receptor for testing. Once expressed in HEK293 cells, CRT1 activity was recorded using whole cell patch clamp electrophysiology. This setup allowed precise measurement of ionic currents across the receptor channel in response to microbe-derived extracts or specific purified compounds. And finally, for the behavioral assays, octopuses were individually placed in behavior tanks and acclimated for 10 minutes before each 60-minute trial. The team used 6 animals, each exposed to different chemical conditions across trials, with both live and artificial crabs presented in randomized order. That sounded complicated. If you're still with me, congratulations. You now speak at least conversational neuroscience. But that was the short version. The full paper is packed with even more technical details, molecular structures, receptor kinetics, and even more experiments that we didn't get to today. If you'd really like to dive into the deep end, you can find the paper, a glossary of terms, and a brief outline of the limitations of the experiments we've discussed today on the companion website. But for now, let's crawl back out of the lab and remind ourselves why all this matters. We start to see the bigger picture. Molecules, microbes, and receptors aren't just chemistry. They're part of a living conversation between an octopus and its world. Next time you think about intelligence, remember the octopus. Their arms aren't just limbs. They are thinking, tasting, deciding entities. Every touch and taste is a conversation with the world and with microbes, gathering information and making truly life or death decisions. This study shows how behavior, sensory perception, and microbial life are deeply intertwined. It's a reminder that intelligence isn't just in the brain, but also in constant dialogue with the world around us. Studying how animals like the octopus sense their environment can reveal entirely different strategies for detecting and interpreting the world. Strategies that challenge our assumptions about intelligence, perception, and decision-making. And that's a wrap for the Octopus Touch. If you're interested in digging deeper into the studies we mentioned, don't forget to check out the companion website, www.mapabrain.com, where you can find links to the paper and a glossary of terms. Don't forget to follow MAPA Brain Lab so you never miss an episode. Science grows through dialogue and curiosity. So drop by the website and leave a comment. I'd love to hear what you learned about the octopus. And remember, the best way to learn science, especially neuroscience, is to teach someone else. So share your notes, invite a friend to join the lab, and we'll discover more neuroscience next time, here on MAPA Brain Lab.